looks well.
Okay, perfect. I'm sitting on a very slow connection here because I'm in the boonies of New York. I actually had some difficulties this morning. I was almost thinking that I wouldn't be able to do this because our electricity went in and out despite the generator here. So hopefully we will not be interrupted. And I'm going to be able to present to you what we have learned about biological control of Japanese knotweeds, which is a complex of two species, Succulenense and Japonica. They are now in the genus Rhinutria. They were before Fallopias or Polygonum, depending on where you look at what timeline. And really, indeed, the update that you will see is of a project that currently only sucks. And what that will mean is becomes a little clearer over time. The focus organism that we will be talking about, I think you can see my cursor here, is Aphalara itadori, a psyllid, a sucking bug that got released from Japan. So I will try to go through this presentation here, trying to manage that. Here's a little timeline of what it is. Basically, what we have been able to do in the US is piggyback on work that was done by CABI in the UK. The lead person, investigator here is Dick Shaw, a little younger at the time that the picture was taken. He has led a consortium that was largely funded by folks in the UK since 2001 that oversees surveys in Japan, and ultimately selected the psyllid Aphalora itadori as an extremely highly host-specific organism as far as they did know at that time. He also rejected many other organisms. There were about 186 in the literature that were located as feeding on Japanese knotweeds in the home range. That's China and Japan, largely, with some outlying populations, probably in Korea and others. And based on some preliminary evidence, he rejected everybody other than Aphalora itadori. They did some host specificity work in quarantine, focusing on some pathogens as well. All of those were rejected because of problematic host specificity results for the pathogens, that is. And then they settled on Aphalora itadori. This was the first project in Europe, actually, where a weed biocontrol agent was actually evaluated. Folks in the US and Canada after that piggybacked on the work that was done by CABI and focused on Aphalora itadori as well. This work was led to a large part by Fritzi Grefstad, funded by the US Forest Service for a long time. And Fritzi is out of the University of Oregon State University. And she has done a lot of the heavy lifting for getting this organism approved. A little bit about what happened. There was a petition filed with TAG. There's a technical advisory group that's advising USDA APHIS after a lot of host specificity results are assembled. That review and that petition for field release was hung up for host specificity results. I can't go into the details here, but it was approved in 2018 by the federal regulatory agencies. There was a Federal Register notice filed in summer 2019. Some of you may know there's a lot of beekeepers that are concerned about losing an invasive species that will feed the bees for a couple of weeks in the fall. That was all addressed, and ultimately approval was given to field release these organisms. Here's a little bit about the field releases that have happened so far. The folks in the UK started to release in 2010, 2011, 2012, and have continued to do so since then. Early on, these were field cage releases, and I will report on that very briefly later. 2018, the first releases were made in Canada, in British Columbia and Ontario. They were faster than in the US, even though the procedure basically was the same. They just went ahead quicker. And in 2020, that's why you're all listening in here. The first field releases were done in the US, again with colonies that and organisms that Fritzi Grafstad created in quarantine in Corvallis, and then they were shipped out to various states. I think I've covered all of them here. There may be other ones that I missed. But basically lots of releases in the Northeast and also in the Pacific Northwest. So that's what happened over the last few months. So here's a little bit about the sucker. Indeed, it's a sucking insect. It's not in the same group as aphids, but it's a plant hopper. It's extremely small. You can see it here on the left side. It's a cartoonish situation. There are 5 nymphal instars, so this is not an insect that goes through a complete metamorphosis like a butterfly would do from a larval, but it's more like a dragonfly, becoming increasingly similar to what the adult would look like. The adults are only 2 millimeters, very, very small. As I age, it becomes more difficult to see them. There are multiple strains that are specific to different Rhinuterus species, we think, but we do not know. We don't know whether there are specific ones that are adapted to the hybrids and how climate may interact with them. We do not know. There's research done right now trying to understand more of that. The releases that were made in the UK, in Canada, and here all came from the same stock that was collected in Kyushu in southern Japan, and so it's called the Kyushu strain. We think it may favor Succulenensis, so that's giant knotweed, which is not particularly common in the East. It's much more common in the Pacific Northwest. The development time from egg to adult is approximately 33 days, depends obviously, of course, a little bit on the life history of the organisms. There are multiple generations, which depends on climate, and it will be region specific. Maybe even specific to specific genotypes. We do not know that yet. We expect, based on Fritzi Grafstad's modeling, that we may have 3 generations here, maybe up to 5 further south in the mid-Atlantic region, if they would establish. The adults overwinter, but we are not entirely sure where. And I blocked my screen. The impact that we expect to see, which is based on information in the laboratory, is a stunting and deformation of leaves. But that has only been seen in the UK quarantine so far. So the releases in New York were done in Tioga and Broome County. I'm not going to disclose this further because we wanted to make sure that we have unmolested establishment at the present time. There's 1 release sites in each county. So that's where it was done. It was released in early June. They were shipped from Fritzi Grafstad's quarantine facility. They were released at long-term monitoring locations. My group, with help from Victoria Nuzu from Natural Area Consultants, has established permanent monitoring locations with funding from the from the Forest Service almost a decade ago, where we have continued to monitor how Japanese knotweeds are growing. These are permanent locations with permanent quadrats. The releases were made about 1,000 adults that were shipped at each site. We kept some in sleeve cages. You will see that in a moment. And we kept the colony on caged plants just to see whether we can learn a little bit about how to identify the organisms, what they would look like, what the eggs and nymphs would look like, and whether we can rear them for some experiments. And then we did follow-up monitoring, which basically requires us to go out into the field with lenses after 1 week, 1 month, 2 months, and then the latest check was in September, just a few weeks ago. So this is basically what it looks like. for the occasion. I was wearing a t-shirt that was celebrating the release of the, excuse me, Gallericella insects on purple loosestrife. Some of you may remember that time. It's a few decades ago. And here you can see the sleeve cages in Japanese knotweed. We released them on the top of the plants. We closed the sleeve bags, and the majority of the insects were actually released in the open. The sleeves are considered to prevent early dispersal, where they would all go away and then not find find each other. Excuse me. The insects were shipped, and here you can see a bunch of them in small vials via FedEx. We shook the curled up leaves with the insects onto a Japanese knotweed plant, and this is just a picture taken immediately after that. Like some aphids and spittlebugs would do, they blow out some residue out of the back end. Those are the white flecks that you see here. There's a specific name for that, which in a moment I forgot. But you have a whole bunch of them just clustered around there, and that was the open release. You've seen the field cages already. This is what they would look like when we were done, or from a small distance on the right. We had 80 to 100 adults per cage. Actually, this is a little less than that. It was per site that should be, so 20 to 30 per individual sleeve, with 5 cages at each site. That, as I said before, prevents immediate dispersal, should allow oviposition and local build-up, at least in the sleeves. And then we check them after 1 week. When we went, yoo-hoo per cage, that seems to be wonderful. No adults were found outside, and very few eggs, which was not necessarily unexpected. And then we removed the cage. So what you would see here on the left side, this is not one of our released organisms, this is just an aphid. But the little white specks here that you see on the underside of the leaf, those are the eggs of Aphalora itadori. A little more clear on the right side. Again, it's difficult to photograph them without a microscope. But we have other organisms hanging around there. Again, these are observations from adults that were caged on sleeves. The eggs both on the underside and on the upper side of the leaves. Well, there's some were dispersed on the underside. On the upper side, they seem to follow the leaf veins. You see a whole bunch of these eggs that turn a little more yellowish over time. When light or the sunlight shines on them, they're really nice to see. And you can see maybe a couple of hundred just in this slide. We did some measurements of what it would mean, where they would oviposit, so that would allow us to then maybe find the eggs easier if there's a preference. And what you see here is, in the graph, is the number of eggs per leaf. The numbers on the y-axis just show you the going from the uppermost portion, the youngest leaves, to the oldest ones. The youngest ones here are not fully unfurled yet. They are curled up, so it's difficult to oviposit. But what you can see, red and blue just reflect upper and lower epidermis of the plant, is that there's no clear preference other than for very young leaves. That's where they like to lay their eggs. That allows the nymphs, the leaves are a little softer, easier access to the tissues that they suck out of the plant. All right, so this looked really great early on, at least for the ones that were released in sleeves. Well, we came back 1 month later, and we found, with maybe half an hour search time at each location, we searched all the stems where we had them released, where obviously the nymphs can't fly, they can only crawl away. What we saw was 1 nymph at 1 site. So that was not particularly great, and rather disappointing, after we probably had something of 10,000 eggs that we are starting with, at least for the ones from the sleeves. So rather disappointing. We did some rearing on potted plants, and so there are 2 other people that are involved in this one. Very clearly, it's Katie Lee, who is a new graduate student with me focusing on Japanese knotweed. If we can do the biological control, you'll hear more about her and what's going on there. And Stacy Endres, who is a postdoc with me, who's both focusing on Phragmites and on Japanese knotweed. They had pets, basically similar to what you see here, a bucket with a potted Japanese knotweed plant, some psyllids in them. Katie said they were making not particular good household pets, not much fun to watch because nothing much was happening, but we had experimental releases on these potted plants where we retained a few hundred adults to maybe potentially use them for additional experiments. It was extremely successful in producing offspring. That was quite clear. Excuse me, I'm getting confused with my videos here. We had hundreds of adults that were produced in these sleeve cages, but the impact was basically non-existing. There was no impact on plant growth that we could discern, even though hundreds of them developed on reasonably small plants that were maybe 2 feet tall. Because some of our insect rearing or our plant rearing didn't go as well as anticipated, we released the remaining adults at Tioga County release site in August. So the summary for the 2020 releases: there was no confirmed establishment at either New York site. The small size made locating individuals extremely difficult. There was a drastic population decline, except when protected in cages. It's highly likely that predation is having a major impact that was suspected at some other release sites, but that needs confirmation. We saw a whole bunch of mites, different mites that were crawling around there, all kinds of other critters that you will find on Japanese knotweed that we typically overlook, but it's highly likely that not everybody just dispersed and we couldn't find them. It could also be that they didn't like the hybrid. Both sites in the 2 counties were hybrids where we released the insects. Yet when we had them in sleeve cages, they obviously were developing fine and having no problem to reproduce and increase new generations. The New York results mirror results from other 2020 release sites in the Northeast and Pacific Northwest. So we're not unique in failing in getting these things established. So to go back to the other releases that were made both in Europe and in North America, the UK releases, despite being repeated for a decade, no establishment. They can build up maybe small population in cages, no overwintering. Remember that I said we have no idea where they overwinter. That's not known. Canada started releases in 2018, no establishment. Can't confirm that there are organisms present. In the US, as far as we know, no establishment. So what's left for Aphalora itadori? Nothing but hope. Maybe they're hanging somewhere where we're not looking. We're obviously not breaking through every little plant and every stem in a Japanese knotweed infestation. Maybe they're somewhere sitting secretly building high populations. So we will continue to do the monitoring, but this requires clearly a reality check here. Even if populations build, there appears to be no hope for impact. And so while the people that initially selected this selected a very, very highly specific insect, but it doesn't appear to be a good biocontrol agent because it lacks the ability to impact plant growth and ultimately demography, which would reduce populations and the spread that this organism, that Japanese knotweed could have. That's, for me at least, not a particular new insight. I have iterated that for a long time. There was very little hope to begin with, but nevertheless, Aphalora itadori was the only organism currently available, so we jumped at the opportunity that we could release the organism in New York and, as I said, hope for the best. So what's my recommendation now? I know there are people that are eager to jump onto the organism, jump into biological control for Japanese knotweed because nothing else works. Do not invest resources into mass production. You can easily mass produce the organism in quarantine or in a greenhouse on sleeve cages. Nothing will happen in the field. We are not unique in that. Only begin to do this unless establishment and impact can be confirmed at early release sites, whether that's here in Canada or in the Pacific Northwest. Maybe they do a little better on giant knotweed, which is not, as I said, not very common in the East. Otherwise, you are wasting resources. So be forewarned. That's not a really good recommendation for people that have been itching to do something for a long time. I recognize that, but we shouldn't be wasting resources. We will continue monitoring at release sites. Hope is eternal, maybe, but when to give up on the failed organisms is very clear. I was never very hot on it. I'm not going to give up within 1 year or 2 years, so we will continue to do that. But the reality check is very clear. My outlook for Aphalora itadori is not very good. However, Cavi is testing a new strain. I talked about that the organism has multiple strains. We have shipped hybrid genotypes just last week to Fritzi, and she's shipping them to the UK to see whether they're going to do better on the hybrid genotypes, the new strain. But the question is whether this is a smart move, because the lack of impact continues to be a concern. Even if the organism does better on the hybrid, will it result in an impact? I question that, but I'm not the person who is dictating what other people are doing. I can only offer my advice, and we have a large group of people from across the US and Europe, with Dick Shaw being part of that, where we discuss how to advance this program, and includes folks in Canada and Fritzi and others. And we will continue to think about what is best as an outlook for biocontrol of Japanese knotweed. So what I say is people rejected organisms early on, but I haven't forgotten about them, and others have not forgotten about them. There's a large number of organisms in Japan and in China that offer enormous hope if they can be shown to be reasonably host specific for biocontrol of Japanese knotweed. So while I have no hope for Aphalara itadori, there is hope for exploring other organisms. And there's a Galarucida here, a leaf feeder. There's a sawfly here in the middle. There is a stem miner, and there is a root feeder. Dick Shaw and the other people in the UK early on rejected them because the literature showed them to maybe not be host specific, but we may have host genotypes. We can have all kinds of specialization, or the feeding that they early on saw makes no difference in terms of the demography of the species that are being attacked. So alternatives do exist. This is not the end of the road for the plant and its biocontrol. So the search for highly specific agents was clear, but they are not effective at the present time if you focus on Aphalora itadori. Small feeding other plants, if it's inconsequential for the plant individuals or for plant populations, that shouldn't be a concern for not releasing a biological control agents. I can't get into the detail, but it's very similar to what we're facing on water chestnut or many other biocontrol organisms. In quarantine and others, they will occasionally attack other plants, but after that, it's inconsequential. We need to focus and restart overseas surveys and work on demography. Now, you have all commented on how difficult it was to work given the constraints and circumstances that we're facing right now. I was supposed to go to Japan and China this year. Obviously, that did not happen, and we don't know whether we can go and do this work next year. That will all depend on how the COVID situation develops, whether we are actually allowed to travel internally or externally, whether we are coming from the US, will be welcome in China or in Japan. So we will see. We have collaborators in these different places that can help us out. We have some funding from through the New York Invasive Species Research Institute from the DEC that was awarded to us last year, and that funding started that we can do some of this work. We will write additional grants. We will do things collaborative. But we need to explore whether these organisms have more than have been rejected appropriately early on by the investigators in the UK. So we will do that. In addition to that, we have a little bit of a science support projects that early on focused on impact of Japanese knotweed. We did some work on green frogs, on plants, on decomposition that has long been published. We've been working on phenology. We have the long-term monitoring at these different field sites to assess growth and potential spread at select permanent locations. And we have a huge joint project, largely funded by German and Chinese collaborators, where we study plant selections that in Europe, 1,000-kilometer transect was done, and Japanese knotweed was collected. The same in the US. The Germans actually came over here and collected plants from Georgia to Maine. We will study all kinds of things from phenology to local adaptation to herbivory. And we will have 3 common gardens, one in the US that has the US plants. This is what it'll be looking like. We have a similar common garden in Germany that has the plant selections from across Europe. And there will be a Chinese common garden close to Shanghai that will host the selection of the US plants, of the European plants, and the Chinese collection that were done by a Chinese team this summer. So that offers us all kinds of opportunities to do additional work. Hopefully Katie and Stacy will be participating in that one. It will be a great jumping off point for doing all kinds of in the Cornell quarantine to understand how our insects would be performing on different genotypes and plant collections from across the climate gradient. We do even little things of trying to do and propagate Japanese knotweed doing stem cuttings, right? It's a weed, it should grow in all kinds of different circumstances, and we can do regenerate that, which is important for us when we send material to China for them to regenerate US plants. So in summary, there is hope. As always, there's little hope for Aphalora itadori. There's lots of hope for the development of additional biocontrol insects for Japanese knotweed. We have some initial funding that will get us going to do an additional checkup. Is it working? Is it not working? Is there more potential, or should we close down this biocontrol program and do I don't know what? So that's the current status right now. All I can say is we're heavily in that one. Stay tuned, and hopefully I have some better news to share with you in a year or two, maybe even from Aphalora itadori, although I doubt that.